Hello everyone, welcome to my YouTube channel. I'm Dr. Malik Siddiqui. As we all know that in 1983, Carey B. Mullis devised a technique which is known as polymerase chain reaction, or in short PCR. This technique has great impact on all the walks of biological sciences, for example in pharmaceuticals, in diagnostics, or in the research laboratories. But in recent years, we hear another term, that is real-time PCR. So, real-time PCR is today's topic. In this topic, in today's lecture, we will try to discuss the limitations of what we call as the traditional PCR or endpoint detection and the real-time PCR. So, my today's topic will be real-time PCR versus traditional PCR. Let us have some introduction about PCR. PCR is an in vitro technique for the amplification of a region of DNA which lies between two regions of known sequence. So it is the in vitro amplification of a particular segment of DNA, the sequence of which is known to us. Now the PCR amplification requires oligonucleotide primers. The primers are basically short and single stranded oligonucleotides which are complementary to the outer regions of the known sequence. So now you can see, so there will be two primers required. One primer will be complementary to the coding strand and another will be for non-coding strand. Now the evolution of this PCR to real time, that is, the PCR has completely changed the detection of RNA and DNA, whereas in traditional PCR, the detection at the end point of the reaction is done, but in real-time PCR, the detection is done while the reaction is occurring. So, traditional PCR has advanced from the detection at end point of the reaction to the detection while reaction is occurring. Now, real-time PCR allows to detect amplification during the early phases of the reaction. The measurement of the kinetics of the reaction in the early phases of PCR, it provides distinct advantage over the traditional PCR detection. In traditional PCR methods, we use agarose gel, and then we will be using staining techniques like ethidium bromide for the detection of PCR amplification, and that is done at the final phase or end point of the PCR reaction. Let us have some idea on the limitations of endpoint PCR. Now, agarose gel results are obtained from the end point of the reaction, so that is after the PCR is done, we have to separate them in agarose gel, and then ethidium bromide or some other dye will be used. So this is endpoint detection is time consuming. So the results may not be obtained for days. And secondly, the results are based on size discrimination, which may not be very precise. While the gels may not be able to resolve these variabilities in yield, the real-time PCR is sensitive, and it is sensitive enough to detect these changes. For example, in agarose gel, resolution is poor, about tenfold, but real-time PCR allows as little as twofold change to be detected. So, we can summarize the problems with endpoint detection: poor precision, low sensitivity, short dynamic range, that is less than 2 logs, low resolution, non-automated, size-based discrimination only, results are not expressed as numbers, and ethidium bromide which is used for staining is not very quantitative, and post-PCR processing, that is, after PCR is finished, then we have to run the gel, most commonly the agarose gel, and then ethidium bromide staining will be there. So that is post PCR processing is also required. Now this you can understand with the figure. So as you can see in this figure, the samples in the gel, they contain 10 copies and 50 copies. So on the left hand side we have 10 copies and on the right hand side we have got 50 copies. So that is after doing the PCR and then running the gel. This is the results which we are getting for 10 copies and 50 copies. Now as you can see, it is hard to differentiate this type of results when there is 5-fold change. 5-fold change, that is 10 copies versus 50 copies. So it is 5-fold change and it is hard to detect on the agarose gel. But the real-time PCR solves this problem and it can detect as low as 2-fold change. That is, for example, if we have two samples differing only in 10 and 20 copies, so this is 2-fold change. This can be easily detected by real-time PCR. Now we have to understand the phases of PCR, because it will lead to an understanding that why the endpoint PCR is limiting. So it is important to understand what happens during a PCR reaction. So the basic PCR run occurs in 3 phases. First phase is exponential phase. What is exponential phase? That is the exact doubling of product is accumulating at every cycle. For example, for PCR we write as the product of one cycle, they are 2 raised to the power n. That is, for example, after one cycle we will have 2 copies. Then as you go on, for example, after 2 cycles we will have 4 copies. So this is the exact doubling of the product. Okay, so the reaction is specific and precise. This phase is exponential phase. Then the second phase is linear phase, which has high variability. So the reaction components are being consumed, because after the doubling, then the reaction components are being consumed, the reaction is slowing and the products are also starting to degrade. So this is the linear phase. Then the last phase is the endpoint or plateau, and then gel detection for traditional methods. Then in the plateau phase the reaction is stopped, no more products are being made, and if left alone the PCR products will begin to degrade also. Now here, now you can see in this figure shows 3 replicates of a sample. Now as you can see this picture, on the x-axis we have number of cycles, on y-axis we are plotting the log of DNA concentration. So there are 3 replicates of the sample. The replicates have the same starting quantity. Okay, as the PCR reaction progresses, the sample start to amplify in precise manner. The amplification occurs exponentially, that is the doubling of product, which is called amplicon. So it occurs in every cycle. This type of amplification occurs in the exponential phase. This exponential phase occurs because all the reagents are fresh and available. Now the kinetics of the reaction put the reaction to favor doubling of amplicon. But as the reaction progresses, some of the reagents being consumed as a result of amplification. So this depletion will occur at different rate for each replicate. Now you can see in the figure, the green part is the exponential part, exact doubling. Then we have got linear phase where some of the reagents being consumed. So now you can see the 3 lines, they are separating. The replicates, they are separating. And as the reaction progresses, some of the reagents being consumed and the results of amplification. This depletion will occur at different rates for each replicate. Okay, understand this point, that in the linear phase we have got 3 different lines and 3 different lines are separating. Why this is happening? It is happening because the depletion is occurring at different rates for each of the replicate. Now the reaction start to slow down and the PCR product is no longer being doubled at each cycle. So in this phase the doubling has stopped. This linear amplification can be seen in linear phase of the reaction. The three samples begin to diverge on their quantities during the linear phase. Now come to the third part, that is the plateau. Now you can see the purple lines, which is the plateau or endpoint. So eventually the reactions begin to slow down and stop altogether to reach a plateau. This is shown as the purple colored. Okay. So in this case the yield will be variable. So each tube or reaction will reach plateau at different point. Why it is happening? Because different reaction kinetics for each of these samples. Now these differences can be seen in the plateau phase. Now you can see three distinct purple lines which show the three distinct plateau phase for three replicates. The plateau phase is where the traditional PCR takes its measurement, also known as the endpoint detection. So in the traditional PCR, after this run is complete, after the plateau is reached, then we will use, for example, agarose gel and stain it with ethidium bromide. Then we can have our results. Now if you see the PCR phases in the log view, this picture, the amplification phases can be viewed differently to assess the PCR phases. This figure shows the phases of PCR in log scale view. On the x-axis we have number of cycles, on the y-axis we have log of DNA concentration. The initial green line shows the area of detection for the real-time PCR, whereas the black line plateau shows the traditional PCR detection. Now you can see the two arrow. First arrow is pointing towards the green part of this graph. That is the detection which is done in the real-time PCR. And now you can see the plateau, another arrow showing the black part of the graph, and this shows the detection in traditional PCR technique. So that is after the run is complete, endpoint detection. Now this is another figure that shows the PCR phases in linear view. Now the PCR phases in the linear view are shown in this picture, and these are in linear scale view. Here the DNA concentration, not the log of DNA, but DNA concentration is plotted on y-axis and the number of cycles on the x-axis. Again the green part is the exponential, that is the first phase, followed by the linear and ultimately the plateau phase. And in the plateau phase, in the traditional techniques, we will do the estimation, we will do the detection, whereas in real-time PCR we will do the detection in the exponential phase only. Now we can understand this thing better by comparison of a linear and log view of 96 replicates. You see these two pictures, the plateau effect. You can see the first picture, it shows the linear view and the other is the log view. So what I am trying to do is to explain the effect, plateau effect, by these two graphs. As you can see that in the linear view the reactions show a clear separation in the plateau phase. Are you able to see this? The lines starting in the exponential phase you are not able to detect, but after some point, after reaching the plateau, you can see clearly the separation of these distinct line. That is, it is of 96 replicates. So clear cut separation can be seen. Therefore, if measurements are taken in the plateau phase, the quantification will be affected. Are you getting this point? That in the linear phase, this linear figure, which is the first figure, if we are doing the detection at the plateau stage, plateau phase, then the measurement will be affected, because now we have got different lines, they are separated. Okay, the other figure, side figure, it shows, this is the log view. Log view means log of DNA concentration. Now you can see in the log view that the plateau of each reaction seems to occur in the same place. Now in the plateau phase, you can see all the lines are merging in the plateau phase. Okay, so it is solely because of the log scaling of the plot. So now you can see clear-cut segregation in the exponential phase. Okay, so in real-time PCR, because we are doing the detection in exponential phase, and therefore the detection is much more easier rather than detecting after the plateau is achieved. This is again comparison. This is the comparison of linear and log view of 96 replicates, same figure. Now to understand it much more better, let us see the log view of 5-fold dilution series. In the log view of 5-fold dilution series, it seems a plateau to occur at the same place, even though the exponential phase clearly shows difference between the points along the dilution series. Can you see this, the graph? In the plateau effect, you see the lines are converging towards each other. Okay, so this reinforces the fact that its measurement are taking place at the plateau phase, then the data would not be truly represent the initial amounts of the starting target material. Getting my point or not? On the x-axis, we have got the number of cycles. On y-axis, we have got the DNA. Okay, and now you can see in the exponential phase, they are different. So detection in real-time PCR is done in the exponential phase. Okay, so the amounts of starting target material, they are different. They are clearly separated. Now the real-time chemistry provides fast, precise and accurate results. Real-time PCR is designed to collect data as the reaction is proceeding. Okay, so because in real-time PCR we are doing the detection in the exponential phase. So from this figure you can clearly understand this point, that when we are doing in the exponential phase, the results can be distinct. So it is more accurate for DNA and RNA quantification, and it does not require the laborious post-PCR methods. Got this point? Now the question arises, how do we do the quantification? Yani during the exponential phase, how we will do the quantification? So theoretically, there is a quantitative relationship between amount of starting target sample and the amount of PCR product at any given cycle number. The real-time PCR detects the accumulation of amplicon during the reaction. The data is then measured at the exponential phase of the PCR reaction. Whereas the traditional PCR methods use agarose gel or other post-PCR detection methods which are not as precise as real-time PCR. As I mentioned earlier, that the exponential phase is the optimal point for analyzing the data. So real-time PCR makes quantification of DNA and RNA easier and more precise than the past methods. Now the techniques for the quantification which are used in the real-time PCR. So 5' nuclease activity, The amply tagged gold DNA polymerase has 5' exonuclease activity. The 5' exonuclease activity of amply tagged polymerase and fluorescent resonant energy transfer makes it possible to detect PCR amplification in the real time. So, one of the methods is the 5' nuclease activity. The 5' exonuclease activity of the enzyme acts on the surface of the template to remove obstacles downstream of the growing amplicon that may interfere with its generation. Okay, this 5' nuclease assay uses this activity in real time detection. If you are not understanding this thing, let us understand 5' exonuclease activity of tag polymerase. Here the polymerase is adding bases to a growing chain of DNA. Subsequently, the polymerase is removing DNA that is downstream and therefore impeding its capability to synthesize the new strand. Here now you can see the new base addition and the base removal. Come to the next. What is fluorescent resonant energy transfer, or in short we call as FRET? The FRET or fluorescent resonance energy transfer technology is utilized in the 5' nuclease activity. The principle is that when a high energy dye is in close proximity to a low energy dye, there will be a transfer of energy from high to low. That is, high energy dye is transferring the energy to the low energy dye. Here you can see. Now again coming back to 5' nuclease assay, how we do the assay. In the 5' nuclease assay, an oligonucleotide called as Tachman probe is added to the PCR reagent master mix. The probe is designed to anneal to a specific sequence of template between the forward and reverse primers. The probe sits in the path of the enzyme as it starts to copy DNA or cDNA, complementary DNA. When the enzyme reaches the anneal probe, the 5' exonuclease activity of the enzyme cleaves the probe, which you can see in this figure. Now another figure, I have tried to explain the polymerase collides with Tachman probe. In the second figure, we have the cleavage of the Tachman probe. Now increased fluorescent activity due to the cleaved probe. Now the Tachman probe is designed with high energy dye, which is termed as reporter at the 5' end, and a low energy molecule termed as quencher. So you can come to the previous slide. This. Now you can see the reporter dye and quencher dye. So the Tachman probe is designed with high energy dye, which is termed as reporter at the 5' end, and a low energy molecule termed as quencher at the 3' end. When this probe is intact and excited by light source, the reporter dye's emission is suppressed by the quencher dye as a result of close proximity to the dye. Okay. Now come to the next figure to understand it better. Here now you see. So when the probe is cleaved by 5' nuclease activity of the enzyme, the distance between the reporter and the quencher dye increases, causing the transfer of energy to stop. Now the fluorescent emission of the reporter increases and the emission from the quencher decreases. Here you can see The green line shows the emission by the reporter, and the red line shows the emission by the quencher dye. So when the probe is intact, you can see the fluorescence, okay, and after the probe is cleaved, then you can see the emission by the reporter, which is green color, it is increasing, and the emission from the quencher, which is shown in the red color, it is decreasing. Now, how this increase in the reporter signal is achieved? Let us understand by this graph. The increase in the reporter signal is captured by sequence detection in the structures and displayed by the software. Now, this figure which I have shown here, it is a graph between the time versus fluorescence, emission of fluorescence. So, this figure shows the increase in the reporter signal over time. The amount of reporter signal increase is proportional to the amount of product being produced for a given sample. Okay, so you can see as the time increases, the fluorescence by reporter signal is increasing. Now let us understand it more better by another curve, which is the amplification curve. On the x-axis we have number of cycles, on the y-axis we have the measure of the reporter signal. Now the combination of FRET and 5' nuclease activity of AmpliTac gold DNA polymerase, it enables the 5' nuclease assay and the STS instrumentation to collect the data in real time. When the fluorescence signal reporter increases to detectable level, it can be captured and it can be displayed as an amplification plot. So this curve is the amplification curve, amplification plot. What is this plot? This is the plot between the number of cycles and the measurement of reporter signal fluorescence. This amplification plot contains valuable information for the quantitative measurement of DNA or RNA. The threshold line is the level of detection or the point at which the reaction reaches a fluorescence intensity above background. The threshold line is set in the exponential phase of the amplification for the most accurate reading. The cycle at which the sample reaches this level is called as the cycle threshold, which is denoted by CT. Now you can see what is CT. It is the cycle threshold, that is at which the sample crosses the threshold level. The cycle at which the sample reaches this level is called as the cycle threshold level, which is denoted by CT. These two values are very important for data analysis using the 5' nuclease activity. Now, what is used is CyberGreen dye. CyberGreen Chemistry is an alternative method. This is alternative method used to perform real-time PCR analysis. This green dye is a green dye that binds the minor groove of double-stranded DNA. When CyberGreen dye binds to double-stranded DNA, the intensity of fluorescent emissions increases. As more double-stranded amplicons are produced, the CyberGreen dye signal will increase. So now you can see detection is done at the exponential phase. So as more and more amplicons are produced, the CyberGreen dye will signal more emissions. The next figure shows the entire process of each type of real-time chemistry. The CyberGreen dye will bind to any double-stranded DNA molecule, while the 5' nuclease assay is specific to the predetermined target. Now come to the applications of real-time PCR. The real-time PCR can be applied to traditional PCR applications as well as the new applications that would have been less effective with the traditional PCR. Now, with the ability to collect data in the exponential growth phase, the power of PCR has been expanded into applications such as viral quantification, quantification of gene expression, array verification, drug therapy efficacy, DNA damage measurement, quality control and assay validation, pathogen detection, and genotyping. So these are some of the applications of real-time PCR. Now, what are the advantages of using real-time PCR over the traditional methods? The traditional PCR is measured at endpoint, that is the plateau, while the real-time PCR collects the data in exponential growth phase. So an increase in the reporter fluorescence signal is directly proportional to the number of amplicons which are generated. The cleave product provides a permanent record amplification of an amplicon. And there is an increased dynamic range of detection. No post PCR processing is required, and the detection is capable down to a two-fold chain. So it's a sensitive technique, much more sensitive than the traditional method. So before ending the topic, please note some of the important things: that the practice of patented polymerase chain reaction, it requires a license. Then the PCR process and the 5' nuclease process are covered by patents which are owned by Roche Molecular Systems Incorporation and F. Hoffman La Roche Limited. Now the applied biosystem is a registered trademark, and the AB design and Aplera are the trademarks of Aplera Corporation and its subsidiaries in the US and other countries. Now the term which I used in my topic, Amplitag Gold and Tacman, they are the registered trademarks of Roche Molecular System Incorporation. Then CyberGreen is a dye, it is a registered trademark of Molecular Probes Incorporation. So you have to keep these things in mind also. And therefore, if you liked my lecture, you can like, share and subscribe to my channel, and I always welcome your valuable comments. So once again, thanks for the listening. Thank you very much.